While I was preparing these lecture slides, I recalled this quotation from John Arbuthnot, 1692, who was one of the first people to actually use epidemiology and statistics in the study of disease. There are very few things which we know which are not capable of being reduced to a mathematical reasoning. And where a mathematical reasoning can be had, it's as great a folly to make use of any other as to grope for a thing in the dark when you have a candle standing by you. I just thought that seemed to encapsulate what molecular genomics offers at this point in time to clinical medicine. It's a complementary technology. It offers alternative insight. And that's what I'm going to be talking about today. Because as we enter the 21st century, mathematical reasoning, which is specifically embodied in molecular biology, has advanced to the point where we know the precise location of atoms in certain key molecules which control the human body. And we can use the genome to predict the location of atoms in many other molecules, and predict with sufficient accuracy to understand the precise interactions between drugs and those molecules. An understanding which is often proven elusive in the clinical environment. And that is in fact exactly what we're talking about here. ARBs, statins, and you would all be aware of the tremendous amount of research that's been expended on trying to figure out how ARBs differ from statins and differ from ACE inhibitors. The molecular genomics can help us understand what to look for when we go back into the clinical environment. So we're talking about atoms. Well, there are lots of atoms in the human body, far too many for us to consider individually. This is a picture from one of the figures from our recent paper, common angiotensin receptor blockers may directly modulate the immune system via VDR, PPAR and CCR2B. Those are all molecules in the human body that have specific functions. And this particular picture might look rather pretty, but it's not very useful for its major purpose. Its major purpose is to show that this ARB here is docking or has a strong affinity for the receptor. There is the rear of the ARB molecule, and here is the front of the ARB molecule. But using a representation like that is not very helpful in terms of trying to understand how these molecules actually work. So in order to make it easier to understand the structure of very large proteins, a representation which highlights helices, folds and flaps has been developed. And we let the computer remember where each atom is located and focus on the overview. The previous slide showed just the upper right-hand corner of this same GPCR up here, but here the ARB in the binding pocket can be far more clearly seen. This protein is the CCR2B receptor, which allows monocytes to migrate to regions of infectious and physical trauma. Also, some HIV strains enter the phagocyte through CCR2B. So it's a fairly important molecule. We can also produce two-dimensional molecular representations, which are extremely useful when we're trying to figure out whether we're looking at an agonist or an antagonist, whether the drug is acting in order to enhance the operation of the receptor or to block operation of the receptor. And here is the same ARB and the same CCR2B binding pocket, but now you've got the detailed atomic interactions. You've got each of the residues, each of the amino acids in the receptor, and specific lines showing which of the ligand or which of the ARB atoms are within a bonding distance, or certainly a van der Waals distance. And here is the hydrogen bond. The hydrogen bonds are very important because they tend to be quite a lot stronger and orient the molecules in the receptor. But in general, you only go to the 2D representation when you're going for extreme detail. It's far too complex otherwise. Now we can also model pathogenic genomes. Here is a protein protein is called SAR-0276, which is a putative membrane protein within the genome of methicillin-resistant Staphylococcus aureus, species MRSA 2522 Staphylococcus protein, and docked into it, or mated with it, bound to it, is a molecule of the ARB olmesartan. In an upcoming paper, which we've submitted to the ISID, Molecular Genomics identify ARBs as a new class of antibacterial. It's all a matter of dose. We show how this ARB can be expected to inhibit the actions of the protein and thereby disrupt the function of the MRSA 252 organism. In this particular case, the way we came across the protein and realized that it was actually a GPCR family was using standard genomic search techniques by homology with CCR2b, which was the molecule I showed you a little while ago, the one that's prevalent on monocytes and causes the monocytes to migrate to areas of trauma. And this particular protein is in the MRSA genome, and its function, we don't know. But if it's ever expressed by the organism, the ARB, if the ARB is present in the bloodstream of the individual, the ARB will go after that particular protein and bind into it quite firmly. Why do I have this here? Well, it turns out that this whole search that's led to the presentation here today started off with some papers back in the early 90s where biochemists that were working on the development of ARBs found that unless they applied a bactericide to kill any bacteria in their tissue samples, that the radio-labeled ARB was taken up by the bacterial organisms and not by the tissue that was under test. And at the time I had no idea what was going on or why this would be the case, and the biochemists didn't care about it. They just made sure they killed the bacteria in any tissue samples before they did their testing. But it was very interesting as to why bacteria would have an affinity for ARBs. And that's really what started me off on this search, and that's why it's here at this point in time. Now, what we've done is a very large computer search using computer servers running Linux and some new software, well, it's not that new, but software which automatically docks ligands or drugs into proteins. We've taken known proteins and we've taken some hypothetical proteins as well where we don't have an X-ray structure, so we can't be precisely sure that these are the correct shape, but we think they are. And then we've taken known proteins, VDR and PPARG, which have actually been photographed with X-ray spectroscopy in order to find out, well, actually not spectroscopy, but X-ray techniques in order to find out exactly where the atoms are. And we've matched up the common drugs with the receptors. And as you can see from this table here, most of the ARBs and statins have some affinity for angiotensin II receptor, which is a GPCR. That's the receptor like we were looking at earlier, a membrane receptor. CCR2b, which is a putative model again, and VDR and PPARG, which are both nuclear receptors. There are some that don't dock, and there are some that dock with quite high affinity. So this would not normally, losartan into VDR, for example, would not normally inhibit the function of the VDR at the concentrations that the drug is normally administered. But other drugs, such as telmisartan into VDR, clearly significantly affect the operation of both VDR and PPARG at normal concentrations. This is millimolar. The graph is in millimoles. So what am I saying? Sorry. It's nanomolar. So with a 25 milligram daily dose telmisartan, for example, it will create bloodstream concentrations that will affect up to around 10 nanomolar affinities. And certainly at the 0.04 and 0.3 nanomolar, there will be very great interaction. Now, there are a number of surprising results from our study. The first thing is, we were amazed to find that there were no accurate molecular structures of the human membrane receptors, the human GPCRs, available. The entire class of drugs, the ARBs, had been built on decades-old foundations, totally in vitro work. Then, when we managed to construct a viable putative model, because we had to go back to fundamentals and say, well, can we construct a model for the angiotensin II receptor that makes sense, that has the conserved regions in the right spot, that binds the molecules that we know, that binds the drugs that we know bind into it, binds them with the correct affinities. That's what we call a putative model. Not verified with X-ray, but gives us something to work from. At that point, we found that the NDA, the new drug application in vitro work, had been done with bovine and guinea pig tissue, and that there were two key binding pocket residues, isoleucine 193 and leucine 205, which differed between the animal genomes and the human genome, thus equivocating, making uncertain quantitative assays. And I've highlighted the two residues that are mutated here. This residue is mutated in Bos taurus, and this residue is mutated in Caviar porcellus. And you can see I've got a candesartan, an ARB bound into the binding pocket, and there is actually an oxygen on the candesartan that is very tightly bound to the interleucine 193 of Bos taurus. One of the things that we found in our study was that we couldn't match up accurately enough, or as accurately as we expected. We couldn't match up the expectations for the binding affinity of candesartan that were listed in the NDA with the binding affinity that we were simulating in our receptor. And then when we had a look and realized that the NDA had been done with Bos taurus, or a protein from the animal genome, we suddenly realized that there is a huge difference in affinity at this point. And that tended to make us feel a little bit more comfortable with our model for the angiotensin receptor. Here is the entire picture of the GPCR, and you can see the binding pocket is up here behind helix 6 and between helices 5, 4 and 6. The candesartan's bound up there. I might add that this is a membrane protein, so most of this region, the central region of the protein, is within the membrane. The region at the bottom here is in the cytoplasm, and the region at the top is extracellular. I might need that laser pointer. Mine seems to be getting weaker as the time goes by. This one, is it ready to go? Oh yes, there we go. yeah, there we go. So this is the transmembrane region, this is the extracellular region, and this is the intracellular and signaling region. But you know, the biggest surprise we had was that the extremely high affinity which the ARBs had for VDR and PPAR gamma, which are nuclear receptors. These are in the nucleus, not on the membrane of the phagocytes, but in the nucleus of the phagocytes. And they're key to the operation of the immune system. So while it was reasonable that these highly flexible polar ligands, the ARBs and statins, ligand is a term for drug, technical term for drug, but while it was reasonable that these highly polar, very flexible ligands might well have a good affinity for other GPCRs, membrane proteins, other than angiotensin II type 1 receptor, we never expected them to have such a high affinity for the nuclear receptors. VDR and PPAR gamma are located in the nucleus of cells and are some of the molecules which cooperate using a complex interplay of dimerization. Dimerization is where proteins bind to each other to form multiple complexes called dimers. With activation by a variety of ligands, and I'll talk about this a little bit more in a later slide, which transcribe genes from the host DNA into messenger RNA. In turn, this RNA will be translated by the ribosomes into protein strands, which are long protein strands, and they are then folded into the final shape, for example, that GPCR shape with lots of folds. They're folded by obviously electrostatic forces, but also enzymes, and there's some feeling that other nuclear receptors are involved in protein folding as well. But these are at the very heart of the genome. All of the proteins produced by the cells come from this DNA transcription process. VDR is the first one we look at, and the correct operation of the VDR is key to both the endocrine and the immune systems. Some functions of the VDR include decreased parathyroid hormone transcription. It lowers high levels of VDR generally correlate with low levels of PTH because it decreases the transcription of PTH. It regulates the toll-like receptor 2 and toll-like receptor 4 expression, and these are receptors which are on phagocytes, and they're part of the innate immune response. In fact, they're key to the innate immune response. VDR regulates them and consequently regulates the response of the body to bacteria. It transcribes CAMP, cathelicidin antimicrobial peptide, and that is an endogenous antibiotic that the body makes which attacks a lipopolysaccharide on gram-negative bacteria. There are a number of endogenous antibiotics. That's one of them, and we know for certain that that is transcribed by the VDR. It regulates the TACO gene, and the TACO gene is associated with Mycobacterium tuberculosis survival intraphagocytic. In other words, how the Mycobacterium tuberculosis, when it's invaded a cell, how it manages to survive phagocytosis. Regulates the TACO gene. It binds the interleukin-2 promoter and therefore transcribes interleukin-2, another key immune cytokine. It also promotes transcription of insulin receptors, turns the DNA, transcribes the DNA into RNA so that insulin receptors can be made. It interacts with cofactors SRC1 and SRC3. Those are steroid receptor cofactor, SRC steroid receptor cofactors, which are inhibited by P65, which is half of nuclear factor kappa B, again, immune system. And we know it's associated also with the granulocyte macrophage stimulating factor, another key immune system function. It regulates TGF-beta signaling and DRIP coactivators. DRIP is D-receptor interactive protein, so a creative name, I think, but DRIP coactivators, all of which regulate cell differentiation and apoptosis. And there's a URL there of a search engine which will specifically search for citations on the VDR if any of you are interested in looking further. There is so much activity in molecular genomics at the moment looking at the VDR. We published a paper not too long ago, mid-January, and recently I wanted to look it up on PubMed. So I'm lazy, and rather than type in the full name of the author and the normal ways I would retrieve my own paper, I just typed in VDR, carriage return. I thought, well, it'll be somewhere on the first page. No way. There have been 40 papers on VDR published since ours in mid-January, at the rate of about one a day. And half of those are on the immune system and the importance of this receptor to the immune system. PPAR is another receptor. There are two forms. Peroxisome proliferator activation receptor is the full acronym, but PPAR affects the generation of liquids and it also of lipids, and it also transcribes key immune system genes. And here's a quote which I thought was rather good from one of the papers that I reviewed. PPAR gamma was originally discovered as a pivotal regulator of adipocyte differentiation, but it's intimately involved in the regulation of expression of a myriad of genes that regulate energy metabolism, cell differentiation, apoptosis, and inflammation. Due to the links with fat cell development, insulin and glucose metabolism, drugs which affect PPAR gamma are likely to profoundly modulate the lipid metabolism. PPAR gamma also modulates the immune system, especially vascular inflammation. And then the other receptor that we looked at initially was PPAR alpha, which is involved in mediating cholesterol ester hydrolase, CEH, which is part of the macrophage cholesterol homeostasis. It also stimulates keratinocyte differentiation, and it attenuates development of hypertension and of oxidative stress, and additionally attenuates vascular complications, and it's linked with insulin and the corticosteroid metabolism. Again, a key receptor for the immune system. The last few slides have described many of the effects of ARBs and statins, which are currently creating surprise in clinical trials. That ARBs and statins affect, for example, diabetes, certainly the complications of diabetes, and also atherosclerosis. So what we did was we decided to take the key nuclear receptors which have got known structures for them, well, ones where we could be quite certain that we knew we were dealing with a real molecule and not with one that we had derived and tested. And they turned out to be the VDR, the PPAR alpha, the PPAR gamma, the GCR glucocorticoid receptor, the MCR mineralocorticoid receptor, progesterone receptor, androgen receptor, and thyroid alpha 1 and thyroid beta 1. There are 4 thyroid receptors, and we just selected those 2 because they were available as X-ray models. Now, what do these nuclear receptors do? Well, they join together in heterodimers, or they couple within the same receptor as homodimers. They couple with coactivators. They are very interdependent, and they are very redundant. If you take out one receptor, for example, if you knock out the beta-1 thyroid receptor from mice, then the mice end up deaf, but everything else seems to work correctly. So there is a lot of redundancy. If you knock out, however, the GCR, the glucocorticoid receptor, the mice don't get beyond gestation. It's one of the reasons we don't know very much about what the GCR does, because the mice never actually are born alive for us to do further testing on. But what these nuclear receptors are responsible for is transcription of DNA genes to strands of mRNA, which are then translated in the ribosomes into proteins. Now, if you want some basic genomics tutorials, there is a simplified set of flash animations at that particular URL, which I've found to be particularly simple to understand and yet quite accurate. So we're now going to look at some simplified 3D animations of these transcription molecules, just enough to give an overview of what the nuclear receptors do and how the corticosteroids, ARBs and statins affect gene transcription. Corticosteroids now, because as we went further and further into this study, we widened out the scope of interest as to what we were looking for. What I have there is a close-up of the skeleton of a GCR homodimer. What that means is there are 2 GCR zinc finger regions here, 2 proteins, and they are coupled together actually through this zinc finger complex. And they're sitting on top of DNA. You can see the double-stranded DNA here. And these helices, actually underneath the zinc fingers it's hard to see, but that helix and this helix are responsible for causing the DNA to, the bonds in the center of the DNA, which are all hydrogen bonds. There are not any molecules here in the center of the DNA strands. Cause those hydrogen bonds to break apart by forces from the molecules here, and that causes the particular genes to be transcribed. Very simplified explanation, but it'll do. Incidentally, if any of you have questions as we go along, feel free to ask them. Wrong one. Try the right one. There we go. Now this is that same complex, but with a different perspective. You can see the DNA at the bottom. In this case, every single atom in the complex has been labelled. We can select a different type of display here. We'll just select the normal ribbon configuration. And you can see your DNA strands, and on here is the receptor that we saw on the bound into there that we can see on the previous slide. And I just wanted to show you the backbone of the DNA, and you can see particularly the gaps here across the center of the nucleic acids. Those are hydrogen bonds that fill those gaps, and those are what are broken by the nuclear receptors as they cause the protein transcription. Actually, the gene transcription into RNA. I don't know whether that's clear. That was the clearest way I could think of showing what these things do. It's not that simple a concept. They're absolutely key to the operation of the cell. Well, here's a graph that I'm sure most of you will be a bit more familiar with. What we have here is a percent saturation on the left-hand axis, 0 to 100%, and drug concentration across the bottom. And we have the normal curves which indicate the IC50, which is equal to the bound natural ligand, plus KD, where KD is a dissociation constant, or minus log 10 of KI. KI values were what were on the other slides. And what I'm looking at is the homologous binding to VDR. In other words, we're not assuming any saturation of either the drug or the receptor. Therefore, it's homologous binding to the VDR with simvastatin versus 1,2,5-D, which is in orange, and telmisartan versus 1,2,5-D, which is in yellow. And I put a note there that the prednisolone KD is very similar to simvastatin, and it will have a similar attenuation of, or a similar displacement of the active 1,2,5-D from the VDR. The reason the yellow band is so wide is because the concentration at the lower end of the yellow band is the concentration of 1,2,5-D in blood, and the concentration at the upper end is the predicted concentration inside the cells. It's quite a bit higher inside the cells, of course, somewhere in the region of 1 nanomolar, 1 to 2 nanomolar. That's been determined in vitro approximately. The KD values were derived from modeling. And you can see that as the drug concentration increases, the displacement of the original ligand, the active 1,2,5-D in this case, drops to 0, which is as you expect. The more drug you take, the more it's going to displace the active ligand and disable, because these are all antagonists, disable the receptor. And that by the time you get to 10 nanomolar concentrations, which are typically what is used, 1 to 10 nanomolar are typically what's used when these drugs are dosed in a pharmacologic application, they have significantly impaired the functioning of the VDR. So here's a table. I was told that FDA loves tables. So here's a table, and on the left we've got the various drugs. We've got the ARBs and then the statins. And over the top we've got VDR, PPAR gamma, PPAR alpha, the glucocorticoid receptor, mineralocorticoid receptor, the progesterone receptor, and alpha thyroid and beta thyroid. I've left off the estrogen receptors and the androgen receptors because honestly they don't change the overall picture very much. And what you can see looking at this table is there are an awful lot of numbers which are below 1. And any of these numbers that are below 1 indicates that that drug is going to have a very significant impact on that receptor at normal concentrations that these drugs are administered in pharmacology. Some of them, like for instance telmisartan, doesn't really affect the thyroid receptors, the MCR or progesterone receptor very much, but it really knocks out VDR, PPAR gamma and PPAR alpha. And same with atorvastatin. It is quite strongly, has a strong affinity for PPAR gamma and alpha, but not too much on the thyroid. But the other thing that's really clear from this is that every ARB and every statin is a little bit different in its activity profile. So whereas clinical medicine looks at a statin as being a statin and really doesn't pay very much attention to whether it's simvastatin or atorvastatin, Lipitor or Zocor, there actually is a huge difference in their profile in terms of what receptors they're affecting in the human body. And but one thing that's common to them all is that all of the statins affect PPAR alpha and PPAR gamma. Some of the statins also affect VDR, notably simvastatin and lovastatin marginally. But that's not unreasonable if you think about it. You've got a drug that's targeted at lipids and it goes after the PPAR receptors. That could be the primary mode of action. We don't know. But certainly the job of molecular genomics is to point it out so that then the in vitro work and the clinical work can go away and say, oh yes, that's true, this is a major function of this class of drugs. And a major reason for the way that they act in which they do. And the same with the ARB. Some of them have a high affinity for the VDR and PPARs. Candesartan, you see, does not have high affinity for VDRs and PPARs. But have a look over here at the thyroid. The thyroid receptors, candesartan has a very high affinity to. And at the other end of the string, you've got something like valsartan, which does actually knock the thyroid.
Can I
well.
ask an FDA sort of question?
Yes.
To what extent do I see the numbers are all over the place. To what extent do we know, let's say, on thyroid function? How much is thyroid function affected when these numbers are low? And we know that there's replacement.
With numbers around 0.5 and 0.7, that receptor is going to be almost totally blocked by the ligand. So now, what the effect of that is on the body is a much bigger, imponderable question, because as I said...
That's what we're interested in.
Yeah.
In other words, can you make that
well, I mean, you can look at it from 2 points of view. The first point of view is that really these drugs shouldn't be doing anything in the nucleus. They shouldn't be affecting the thyroid, for example. So that's the first observation you can make. And then the other observation you can make is, well, the people are sick. They need a drug. So where is the middle ground? How can we select a drug that...
Well, no, if we know the thyroid is being affected, there are medical steps that can be taken. But so that a person who knows that giving this drug will have this effect, we can take amelioration steps.
Yeah, absolutely. You can supplement thyroxine, for example.
We have to be sure that we're not supplementing a person who doesn't need the supplement.
Well, I think my major message, the major thing that's in my mind, is that we need to be measuring and looking. So when somebody is being given candesartan, for example, we should be measuring the T3, T4 and TSH. And if it needs supplementation with T4, with thyroxine, then that might be a good trade-off. I mean, that has to be judged by the clinicians as to what trade-offs are there.
Was this assay done in membranes or cell, whole cell?
This is mathematical. This was done in the computer. No in vitro work at all. Okay, so what this needs to be done, what needs to happen now, is these numbers, which are very hard to determine in vitro and determine in a clinical environment, these numbers have to be kept in the back of our minds as we go back to the in vitro work and the clinical work and figure out what to measure, what to look for in order to elucidate the function of the
what clearly
drug.
stands out is that some of these compounds here are water soluble. They don't have binding into the extracellular membrane. Neither are they transported into the cell. One of these examples which stands out in the statins, you can see that unless it's in the liver hepatocytes, it doesn't go into the cell at all. But I could see numbers very impressive in nanomolar range. It just makes it hard to understand.
Well, I can tell you for certain that olmesartan directly affects the VDR, because I have seen clinical data which indicates that the levels of 1,2,5-D and PTH and secondarily the thyroid hormones react to administration of olmesartan. And yet in the NDA, olmesartan is listed as not having any permeability through erythrocyte membranes. So frankly, I think that's wrong. So I think that what we need to do now is go back and figure out, well, how much is getting in, if any is getting in. I mean, let me put it another way. You're telling me that olmesartan can't get into the phagocyte, and yet HIV can? Do you understand what I'm saying?
Yes.
It just doesn't stand the taste test. It just doesn't sound right. And as I said, with olmesartan, we know because we've got some anecdotal clinical data. That's why we looked at VDR, because it was reported that when people started to take olmesartan, that their 1,2,5-D levels plummeted. In one patient, it dropped in half within 2 weeks. So that's why we looked at VDR. I mean, you can look at other drugs into these receptors and they don't bind. But the ones that I'm showing you on these slides generally have an affinity, and that's why they're on the slides. The 99.9% of the computer runs that we did, which didn't come out with an affinity, are not on the slides. Does that answer your question? But yes, that's a very good question. But on the next slide, there really is a lot less doubt. These are all nuclear. These are all drugs which are active in the nucleus. The steroids are active in the nucleus, and so we know they're there, and we know all of these are active in the nucleus. And we can see some very interesting things that are coming out in the that the mathematics is telling us in the genomics. We can see that, for example, 1,2,5-D3, which is the active ligand for VDR, has actually 10 times higher affinity for the beta thyroid, and similar for the alpha thyroid. And that lines up with anecdotal indications we have that, in fact, high levels of 1,2,5-D in sick people does affect their thyroid function. We can also see that cortisol, for example, has a different profile from prednisolone. For example, into PPAR gamma, there is a significant difference in affinity, whereas into the VDR, they're about the same. Same with PPAR alpha, there's a significant difference, and there's not much into the alpha thyroid, 2 to 1. The amount of error that's involved in the molecular genomics calculations is quite high, and you get anywhere within 3 to 1 or so, and the numbers are essentially, you have to regard the numbers as being essentially the same. Dexamethasone also has a different profile from prednisolone, which is interesting, and that could be clinically useful if it's kept in mind as clinical trials are performed. But you can see that dexamethasone to cortisol is somewhat more accurate, except in the thyroid receptors. It's somewhat more following the profile than prednisolone is. And then for completeness, I've shown the corticosteroid vitamin D3 at the top, which is also active in all these receptors. And as you can see, that also hits the thyroids pretty hard. Well, this was the question you were asking. What does this mean to the FDA? Well, just as John Arbuthnot said, if you've got some information, use it. And use it to try and understand the bigger picture. Because there has been so much effort put into trials, clinical trials of ARBs versus ACE inhibitors. And if the molecular genomics had been applied at an earlier stage to give insight into what to measure in these trials, what sort of endpoints could we expect, then a lot of inefficiency and also the answers would be coming up a lot more quickly as well. By predicting the biological molecules which a particular drug is going to target, obviously the clinical researchers can be guided as to what side effects to look for if they're unwanted, or as to what metabolites to measure in order to get the best overview of dosage and efficacy profiles. For example, I said there's one publication a day on the VDR, and yet I would doubt that there are very many applications before the FDA at the moment that even measure the metabolites that are affected by the VDR, and yet it's the key immune system receptor. It does key functions, and certainly is key to any infection capability that the phagocyte has, anti-infective capability. We need to watch these things. If a drug hits VDR, PPA gamma, and PPA alpha, the clinical trials need to be told, be aware of infection, log infection. Additionally, in vitro testing is not very selective. You can add a drug to a cell line that might affect dozens of different metabolic pathways at once. And the art of the expert, of the perfectionist in vitro, of course, is to stop this and to try and focus in on the one metabolite that you're interested in. But it doesn't always work. In our paper, we looked at a study which had shown that telmisartan was a partial agonist of PPAR. And we pointed out that we felt that it was far more likely that telmisartan was affecting VDR, and since VDR wasn't being monitored in this particular in vitro experiment, that was why they were seeing the specific results that they got. Modelling is very precise and will isolate likely effects to the level of the individual molecule. But the two techniques are complementary, each reducing the need to grope about in the dark, as Arbuthnot says. They have to be viewed as complementary. And I think the example you gave of do these drugs even get into the nucleus is key to that. I mean, it has to be viewed from both points of view. And if you assume that the drug doesn't get into the cell, either through the outer membrane or the nuclear membrane, then the clinical trials will be looking for a different set of metabolites. But if you think there's a possibility it might get in, you can measure the effects that it would have. And if you see those effects, you know that it is in fact getting in there and modulating the metabolism. Now let's look at two example case studies. These were just a couple of drugs that came up while I was preparing this presentation about a week and a half ago. I was watching an advertisement on TV which showed some persuasive animations purporting to demonstrate how the drug ezetimibe, Zetia, reduces the absorption of fat from the GI tract. You've probably all seen these television advertisements. And since I know that the lipid and cholesterol metabolism, much of it traces back to the VDR and PPAR, it occurred to me to look at the NDA. And when I looked up the NDA on ezetimibe, I found unknown mechanism of action. So we ran a scan on ezetimibe. And the next day, Reuters carried a news article about a study with rimonabant, which the study, if I recall, had caused an average of 15 pounds of weight loss in a year in a particular cohort. And I also took a quick look at that drug as well. Now, rimonabant is actually fascinating because if you were designing a drug to target VDR and PPARs, this would be the drug. You can see that it has no effect on the other nuclear receptors. This 72 is negligible at the dosages used. And it has a very significant effect on PPAR alpha in one of the isomers, and the other isomer has a very significant effect on PPAR gamma. And moderate effect, this isomer effect on VDR is moderate, probably wouldn't show up at the 25 milligrams used in the trials. Similarly, for ezetimibe, just like the statins, it affects the PPARs and affects them fairly strongly. 7 nanomolar is a fairly strong affinity at a typical dosage for ezetimibe, which is also in the 25 milligrams a day region. Isomers. Those of you that know what an isomer is, please bear with this. I just wanted to go over a quick slide to show what isomers are. Isomers are when you have two configurations of a molecule. And this is the molecule for rimonabant. And if I rotate the thing around. Okay, so I've now rotated this backbone here so that the oxygen is facing towards us. The backbone is rotated around. And you can see in one case, this benzene ring is with the two chlorines is on the bottom. And in this case, a benzene ring with the two chlorines is on the top. And if I rotate it, you can see that where it is all depends on the angle with which it's bound to this nitrogen. There is a hydrogen on the nitrogen as well, which is not shown. You normally drop hydrogens out of molecules at the molecular genomics level because you get lazy. The computer knows where the hydrogens are. But there is a hydrogen on this nitrogen, and if it changes in location, then you get an isomer. It's the same drug, but it's a different shape. There are many enzymes in the human body that can change drugs from one isomer to the other. The classic case, of course, is thalidomide, and I'm sure you've all been through thalidomide, where one of the isomers, the one that we didn't think existed in the human body, turned out to be pterogenic. Now, rimonabant does, in fact, go into the cannabinoid receptor. It's a cannabinoid antagonist. And I've shown that alongside the agonist, which is called Win55212-2, which is a cannabinoid receptor type 1 agonist. You can see that they're not lying in the same general location. They act on different residues, and rimonabant is an antagonist, exactly as is stated in the press release from Reuters. Except that the interesting thing is that it has about the same affinity for this receptor as it has for the PPARs. Come on. So I wanted to finish here by making a suggestion as to where we can go from here. The last slide, which I'll go back to, the last slide, this cannabinoid receptor was supplied to me by Tiziano Tucinardi, who's a PhD student at Pharmaceutical Sciences in Pisa, Italy. And he'd published a paper, the PubMed ID link is there, on the cannabinoid receptor, and they had produced a putative model and tested it fairly extensively. So I wrote to him and said, can you send me one of these receptors, because I've got this drug that I want to try in the cannabinoid receptor to see what it does. So he sent it to me, I tried it, it docked in there exactly as advertised. But it shouldn't have to do this. It shouldn't have to track down these receptors. These receptors are key to the operation of the body. They're key to the operation of drugs. And why can't we have a database with at least the receptors, which are part of the human body? There's no copyright on them, there's no patent rights on them. Well, mostly there's no patent rights on them. Why can't we have a library of these receptors, just like the RCSB structured data bank, so that people like the student in Italy that I got that cannabinoid receptor from can deposit the receptor, and other students elsewhere in the world can take these and do what we have done with our studies, show that, in fact, there is a significant spectrum of activity beyond what current medical knowledge portends. There's a significant body of information that we could be using the actions of drugs in the human body. The RCSB databank is supported by Science Foundation and a whole stack of NIH and other bodies, including DOE. And I was going to suggest that maybe some of these organisations might join the FDA to help gather together a data bank which would allow students and scientists to more easily study the actions of the pharmaceutical drugs. So much research energy has been expended on the ACE inhibitor versus ARB controversy, and better research data would have hastened resolution. Such a database would also allow rapid analysis of reported side effects and unexpected drug interactions, so that when a side effect is reported that appears way off the wall, we can go back to the molecular genomics and say, well, is it that unreasonable after all? In fact, I think today there is discussion of an MS drug which has an unwanted side effect of infection, or allowing infection. And if those drugs were screened against a known set of receptors and enzymes which are known to be involved in the immune system, it would give us a very good starting point to work from for the clinical trials. So I see that it's 4 o'clock, and I'll be staying after the presentation to chat, and I'd love to speak with any of you that are interested in this topic. And I'm also going to be here tomorrow morning, Wednesday the 8th, and I'd love to talk with groups or individuals at that time that might be interested in looking at these issues in more depth, or maybe just looking at the issues in the same depth, but looking at them again. So thank you very much for coming, and I guess that's it. Thank you.
How did you choose the, what's the end point for the immune system in your case? Or why did you say that these ARBs and statins might be the immune system?
Why did I choose the immune system? Well, we knew that that at least one of the ARBs targeted the VDR, or seemed to target the VDR, possibly targeted the VDR. And because the VDR is key to the immune system, at that point we started looking a bit more widely into its actions. And that's really why we were talking about the immune system. The immune system is, in any case, very closely intertwined with the lipid metabolism, the cholesterol metabolism in any case. It's very hard to separate them out. But I guess what I was surprised was that these drugs that one wouldn't expect to have any effect in the immune system at all did, at least on the computing level, have a very significant effect on key immune system receptors.
Have you looked at biologics?
No.
Are you planning to?
The biologics are pretty large molecules. It's an extra degree of complexity to try and model those. Yes, I have started to think about particularly TNF-alpha.
Because that would give you, what would give you some precision, I think, to begin to develop your thesis here.
Yeah.
Because those are, for the most part, very targeted therapies, as you well know.
Yeah.
In contrast to these drugs, which tend to be dirty.
Yeah.
And it would help you to figure out where the modeling could fit in terms of the cart versus the horse, maybe.
Well, I think it's important to know that these drugs are dirty, for a start, because I think that the average clinician out in the field has no concept that the drugs are dirty. So it is actually important to know that the drugs are dirty.
A lot of things.
Well, yes, but that's the whole job of the scientist, is to get the word out.
But I would encourage you to do that, because to me, I mean, that seems to be the next level of.
It is. It's an extra.
That needs to go on here.
You're dealing with much bigger molecules.
Understood.
There are some issues with the way that the modelling is done. Typically, we work on the receptors as being a fixed receptor, and no protein is fixed. It varies. The position varies a function of time, as you would know, under not only van der Waals forces but hemodynamic forces and other things. So, and when you're dealing with a larger protein, the modelling of the rings and the deformation of the rings becomes very significant, as it does with the steroids. The steroids are devilishly difficult to deal with because you've got the 4-ring conformation there that you have to deal with.
But then, looking then and going to the next iteration, we'll be looking at various genetic SNPs, whatever,
yep.
of the various pathways in TNF, metabolism and such. Because that's where we're trying to figure this out. I'm a rheumatologist. We're trying to figure this out now with the TNF inhibitors and all the biologics that have been released.
TNF is
we think that they're going to do something, and then we just can't seem to find it. For example, so.
I've got this feeling. It's not a hypothesis. It's just a feeling that there's something somewhere that activates all these GPCRs in extreme infection. Everything goes wrong. The eyes, the everything. Don't know what it is. TNF alpha is obviously a good place to start looking. Also interferon gamma, some of those. And yes, we do have that on the books, as it were. But it's an extra level of complexity beyond where we're at now.
Understood.
We've just mastered the steroids, and we thought that was great.
Great, thank you.
Thank you very much.
Is there a simple reference that goes through on a superficial way of the algorithm that was used in your computer estimation of KI?
In our paper, the actual KI, yes, there is a paper that was published by the group at Script that wrote that software that goes into the KI in some detail.
I'll let you give me the reference if you have
I don't have it in my hand, but I can certainly email you with the reference. But in fact,
you're used to this, aren't you?
let me just show you.
I'm beginning to realize that.
One doesn't think of it that way, though.
No, I know, because of history. What I was going to show you that here, this is a vitamin D. This is the steroid prednisolone. Let me get it the right way up. Come on, you stupid thing. Let me do it on a computer screen. I can see it better here. Okay, there's the steroid rings. So here are the 2 steroid rings of prednisolone, and there's the methane on top of it. And here are the other 2 steroid rings, the other 2 parts of the steroid ring. And these rings are all bound together. And one of the things about the steroids that makes them effective is this is a very rigid structure. So it only fits in a few places. It doesn't fit in an angiotensin receptor, no matter how hard you try. Now, if you take vitamin D, and this is pure vitamin D, it's not 125D, you can see the same 2-ring structure here, and the same structure at the bottom here, including the methane on the pole. But the difference is there is no bond across here. These 2 atoms are not bonded. That's the only difference between them. Now, from the point of view of molecular affinity, that increases the affinity of this molecule immensely, because these are all rotatable bonds. So it can twist and turn and get itself into those receptors very, very easily.
I really like the examples you gave because they did make things clear. Yes.
Good.
Yeah.